I'm DeAnn Presley, and along with my co-author Peter Tomlinson and our former graduate students Savannah Crossman, Laura Starr, and Catherine Stewart, we'd like to talk to you about our Dynamic Soil Properties Research Project, which took place on the Keith Soil Series, which is a benchmark soil in Kansas. The Keith series is classified as a fine-silty mixed superactive mesic Aridic Argiustolls. 80% of the acres are in cultivated cropland and 20% is in native range. The Ogallala Aquifer underlies this region, and for the fields that have irrigation, it makes it an exceptionally productive soil. So if irrigated, the most common crop rotation is not a rotation at all. It's corn, corn, corn. And because of all that crop residue, strip tillage or even conventional tillage is very common. If it's dry land, however, the most common rotation would be wheat, grain, sorghum, or more recently, people are starting to grow corn instead of grain sorghum, or even some soybeans. But no-till is by far the most prevalent tillage practice because of the moisture conservation. So, are there resource concerns for this key series? Let's look at some issues. So, this is the High Plains Aquifer, which is comprised of the Ogallala Aquifer in the western part and the Equus beds in the central. So, the Equus beds are more shallow, whereas the Ogallala Aquifer wells into these triangles are showing irrigation wells. Most of these irrigation wells will be 300 to 400 feet deep. So this is a map of the estimated usable lifetime for the High Plains Aquifer in Kansas. And you can see the legend over here. It says ST is already at the minimum threshold, or under 25, 26 to 50, and so on. And these are years. Years for how long this is estimated to be usable. And what usable means is that you need about 400 gallons per minute to grow a crop. That's so that the irrigation center pivot can get around and irrigate the crop so that it can both survive and produce its maximum yield. So there tends to be haves and have-nots depending on where you live and where you farm. So if you are in one of these brown areas, you've already lost a lot of water. But if you are in one of these yellow areas, you have a lot of irrigation water. Another issue is wind erosion. So this is, a picture taken from the 1930s, and this is to show you the approximate area where we're going to be is in northwest Kansas. And this image was taken in 2020 by an extension agent, and this is near Russell, Kansas. So wind erosion is something we still fight in this region, and usually for the period of November through April every year, depending on how much cover is in the field and just wind and climate that we have, but the weather that we have that year. But it's still a huge issue, and we've seen dust storms that look even worse than this one. So our objectives in this project, we're comparing Keith pattons of 3 different management systems. So one system is long-term no-till with either cover crops in the rotation or a diverse crop rotation. Diverse crop rotation in our case would be where people are not just growing wheat and grain sorghum, but have added some other crops such as winter peas or spring peas for grain. Another system would be conventional tillage, and then we also have native prairie remnants. And we chose, instead of going with irrigation, we decided to go with dry land because finding irrigated no-till itself is uncommon. And in our region, no one irrigates pastures. And I should say the annual rainfall is between 18 and 20 inches. So 3 management systems, 3 pattons per field, times 3 fields of each kind of management system was the goal. And we tried to sample this all in a 2-week period so that we didn't have time or drastic differences in moisture affecting our results. We wanted it all to be as similar as possible. And we did a suite of physical, chemical, and biological properties to a depth of 1 meter. And we split by depths for 0 to 5 centimeters, 5 to 10, and then 10 centimeters to the remainder of the A horizon, and then after that by genetic horizon. And we also did producer interviews, and we did soil health assessments using our Kansas card that we got from the Kansas NRCS. And we did the soil health assessment involved things like looking for earthworms and penetrometer measurements and things like that. And so this is Peter, and he's running the Cornell Sprinkle Infiltrometer, and one of the items on our soil health assessment card was soil smell. And so this is Savannah, who was our graduate student, smelling one of the sites. So these are where our site locations were in our state. And so Sheridan, Thomas, and Rawlins County were our 3 counties that we sampled in. So key soils, this is a map, again, showing these. This is Cheyenne County is here, and that's the corner county. So zoomed in, and you can see the Keith consociation or Keith in a complex. So for Rawlins, Thomas, and Sheridan counties, there is a lot of Keith mapped there. It's a predominant series, to be sure, for Thomas. So an example of how we organize this is one of our sites. This is a field that's been in no-till for 20 years, diverse crop rotation. So by the way, we're calling that our soil health system. And these clustered down here are in a native prairie remnant that was in the corner of that field. And same landowner in this instance. And our conventional tillage site was just off of this map, 1 mile over, but the same mapping unit, same Keith series, and same slope position. We try to keep everything as uniform as possible except for management. So now I'm going to move into the results for aggregates. So we had 2 different methods. So the first one I'll talk about is the ARS method, which is multiple sieves on a machine. So we had no pre-soak time and 5 minutes of agitation on the multiple sieves. And so at the end, you calculate the percent aggregate stability, and this is for the 0 to 5 depth. And by the way, that's what we're going to present today, is just our 0 to 5 depth. So for rangeland, we had 90% stable aggregates. For our soil health systems were 85%, and our conventional systems were about 75%. And you can see the statistics here. The rangeland and soil health were statistically similar, and both were higher than the conventional. So that's great. That's kind of what you hypothesize you might see. And this is the mean weight diameter, so the average aggregate size using, again, same method calculated here, but the rangeland was 1.97 millimeters versus 0.68 millimeters for soil health versus 0.44. So here you can see statistically rangeland separated from the 2 crop systems. And an additional method was the Kellogg Soil Survey Laboratory method, which is a single sieve method where you start off by forcing all of the aggregates through a 2 millimeter sieve and catching them on a 1 millimeter sieve. So the aggregates are manipulated a lot more. And you can see with this method, our rangeland was 67.6% stable versus 8 for the soil health and the conventional. So I find that to be really interesting, how different our percent aggregation is between the Kellogg Soil Survey Laboratory and the ARS methods. Just the size of those numbers is quite different. And then also the fact that the treatment differences they picked up were they organized a little differently, right? So I find that very interesting. And they've values were very different, and they yielded different results when comparing the 2 treatments. And so my early opinion on this is that I really think this multiple sieve method is superior. I think starting with larger aggregate size in general is superior. So I'm a little more comfortable with that method, I guess. And also soil organic carbon. This is traditional soil testing lab measurement. And I'm just showing you for the 0 to 5 depths here. This is our percent soil organic carbon. And rangeland was 2.94 versus conventional was 2.15 versus the soil health system was 1.79. And this wouldn't exactly be what we expected, but I wanted to make sure to say that it's way too early to make any conclusions because I have not yet used the depth and bulk density to calculate the soil organic carbon stocks to a depth of 1 meter. And I also want to think through and calculate what is the thickness of the soil above where the carbonates come into the soil, the depth. How much mollicapipedon thickness have we lost, if any, with cultivation? So I don't want to make too much of a deal out of these results quite yet. And with that, I'm going to turn this over to Peter.
So moving into some of our biological measures for this project, the first set we'll look at here is soil respiration and permanganate oxidizable carbon. So these are both laboratory measurements. Respiration looks at the production of CO2 upon rewetting of soil samples over a period of time. And similar to what we were seeing for some of the physical properties, we see a separation with the rangeland being significantly different from our conventional and soil health treatments. And we see the same, well, a similar trend for POC. Again, rangeland is significantly higher than our soil health treatment, and we see some similarities between the conventional and rangeland. This is something that's going to need more explanation or exploration. It could have to do, POX or permanganate oxidizable carbon is looking at that most available pool of carbon. These sites were all sampled post-harvest in the fall, and so it's possible that with that conventional treatment, we're seeing some residue decomposition that may be contributing to that as an effective tillage, but that'll need more exploration as we look at this data further. In terms of enzymes, we looked at a suite of enzymes. And why are enzymes important? Enzymes are the catalyst for breaking down organic compounds and releasing inorganic nutrients. And so beta glucosaminidase is involved in the breakdown of chitin. Certainly is connected to decomposition of the fungal component of our soil microbial biomass. And what we see here is this is the first place that all three treatments are significantly different from each other. Rangeland had the highest amount of beta-glucosaminidase, followed by our conventional treatment, and finally our soil health treatment. And beta-glucosidase is also involved in the breakdown of carbon compounds, similar to beta-glucosaminidase. And here, what separates out is, again, rangeland had the highest amount of beta-glucosidase, and we did not see a significant difference between the conventional and the soil health treatments. And so, this gives us a sense on the carbon cycling enzymes in the system. We also looked at a suite of three enzymes associated with phosphorus cycling. Alkaline phosphatase looks at phosphatase enzymes associated or prevalent under alkaline soil conditions. Within a soil, we can have a wide range of pHs, and so given the pH, we may see different trends or different predominances of these pH-sensitive enzymes. Both the alkaline phosphatase and the acid phosphatase are thought to be closely associated with the microbial community and are produced by the microbial community. The acid phosphatase, however, is also, there's evidence that it's produced by plant roots. And so alkaline phosphatase, potentially gives us a look at microbially produced. Acid gives us both plant and microbially produced phosphatase. The alkaline phosphatase is done under laboratory conditions at a buffered pH of 11, and the acid phosphatase is done at a buffered pH of 6. And, you know, again, similar trends that we've been seeing. Rangeland is separating out from our two production systems. Uh, kind of similar to what we saw um, for the for the respiration, or not the respiration, but for the permanganate oxidizable carbon. For acid phosphatase, the rangeland and conventional systems were not different from each other. So that's interesting that we're seeing that similarity here, um, and something that we'll need to look at. Phosphodiesterase is another enzyme associated with the breakdown of organic phosphorus compounds. And in this one, again, we see that trend where the rangeland system had significantly higher phosphodiesterase activity compared to the conventional and the soil health treatment. So, you know, pretty consistent that our rangelands is separating out from our production systems. So we also wanted to look at the microbial community in depth and look at the community itself. And so we used phospholipid fatty acid analysis as a tool that would allow us to get at some questions around not only the size of the microbial community, but also the structure of the microbial community. And so the first values that are presented here are the total weight, or the think about that as the microbial biomass size in these systems. And we can see that again, the rangeland is separating from the conventional and the soil health system. And in fact, the rangeland is about twice what what we measured for our conventional and soil health system. And so with PLFA, we can also look at some specific groups. So I've pulled out for this presentation our buskal mycorrhizal fungi subcategory, and we can see here that the rangeland had significantly higher AM fungal biomass compared to our conventional and soil health systems that were not different from each other. And in a rangeland, it's not surprising that that we're seeing this contrast. You know, fungal biomass tends to be higher or greater in systems where there hasn't been disturbance, and so that fungal hyphae network is allowed or able to become really well established and grow. We also, I've pulled in the gram-negative and gram-positive bacterial categories. So these get at two key bacterial groups of organisms. And again, we can see this very similar trend where the rangeland is significantly higher than both our conventional and our soil health systems. Um, the conventional and soil health are not significantly different from each other, and really, are quite similar in terms of values that we're seeing here. And so, starting to bring this to a close, the major, finding here so far at the near surface is that, our rangeland is quite different from the two other systems that we looked at. We do, see some differences between the soil health and the conventional system. Uh, and I think those are important that we're seeing those. But it also highlights that, even though the no-till fields, with diverse either cover crops or diverse crop rotation, these have been in no-till for 20 years. Cover crops or that diverse rotation integrated into the cropping system over the last 9 years. But we're still not picking up major changes, and so I think this highlights that the semi-arid environment, high evaporative demand of the key of the crops that are well, high evaporative demand of the high plains, all of these come together and impact how quickly change happens. When we think about the longer management history of conventional tillage that predated the conversion 20 years ago. And so, how long this takes, and will these cropping system will the cropping system ever reach the rangeland? That's a question for future research. And so with that, we certainly want to thank NRCS for funding this project and providing the opportunity to work on the Keith Benchmark Soil Series. We had some great landowner partners that allowed or were patient through interviews and allowed soil sampling and heavy equipment to travel across their fields. And then also want to thank the NRCS soil scientists who helped with the sampling throughout this project. And with that, we'll wrap up this presentation.